Good evening. Welcome to today's session, half an hour session of Neet Neurolog series day 9. So, are you all ready with your paper and pen? Shall we start the session? Forgive me for being few minutes late. It's due to a power cut issue. So, we'll refresh yesterday's session. Before that, we have to remember our goal. What is the short-term goal? We have to concentrate completely here for the next half an hour without any distraction. So to yesterday's class in code word series under block 1 we studied about scientist and discovery. If you are able to recollect yesterday's session, type it in the comments. Okay, this one is, I think, students please hold on. That's the last week's session and here it is. So in block 1 yesterday we saw scientist and discovery. Under scientist and discovery we studied about Golgi apparatus in the year 1898 by Camillo Golgi. Ribosomes 1953 George Palade. Because of him they are also called as Palade particles. So the other name for ribosomes is Palade particles. What is the shape of this ribosome? They are granular structures, and Golgi apparatus, they are reticular structures. Okay, next, so here we studied one extra point, ribosomes. If the ribosomes are attached to the endoplasmic reticulum, it is called as rough endoplasmic reticulum. If there are no ribosomes, they are called as smooth endoplasmic reticulum. Next, about the plasma membrane or cell membrane. In 1972, Singer and Nicholson proposed a model called as fluid mosaic model or quasi-fluid membrane, which says that in the ocean of lipids, the proteins are floating, okay, laterally. There is lateral movement of the proteins. And we saw about plasmid, very important for the NEET exam. Under plasmids, we divided it into two sections, that is of bacterial importance in class 11 and in biotechnology class 12. What are the code words used by the NTA to ask about plasmid? First one is extra chromosomal DNA. Second, autonomous replication. This is origin of replication. And what is the third point? Phenotypic character, physical trait, antibiotic resistance, cloning vector, which it acts as a molecular vehicle, selectable marker, it can be asked as a match the following. They are asked as small circular naked DNA present in the cytoplasm. So these are the 6 code words which indirectly represents plasmid. Next we saw about genomic DNA versus extra chromosomal DNA. What is the other name for extra chromosomal DNA? Plasmid. Genomic DNA is the one which carries the blueprint of the cell. It is a single chromosome circular DNA, and the region where the genomic DNA is present, you call it as nucleoid. This is seen in the prokaryotic cell. Plasmids are characteristic of prokaryotic cell, particularly a bacterial cell. They are in a prokaryote. Nucleus is absent, isn't it? All the membrane bound organelles are absent in a prokaryotic cell. So there is no nucleus, no nuclear membrane. The circular DNA is present in the cytoplasm. That region is called as nucleoid. Genomic DNA is very important for the life of the bacterial cell. Here, this extra chromosomal DNA is present outside the genomic DNA. It is a small circular DNA. It gives a survival perk, which means additional character which enhances the life of the organism. Bacteria, example is antibiotic resistance. One expression of the phenotypic character of plasmid is antibiotic resistance. These are the code words: phenotype, physical trait, antibiotic resistance. Okay, then this comes in biotechnology. It is useful in to monitor bacterial transformation with foreign DNA. These all we studied yesterday. If you are able to remember any of these points, type it in the comments, students. And today is day 9. So before going into this session, I'm going to ask you a question. Okay, I'm going to ask you a question. Chromatin, are you ready with your pen and paper? Shall we start writing? Okay, so this is the question for you now. If you know the answer, type it in the comments. Chromatin versus chromosome. Question is, are both same or different? Do you know the answer? Type it in the comments right now. Are chromatin and chromosome the same or different? First question. Second question is, when you can see chromatin and when you can see chromosome in a cell. Okay, in a cell, when you can see chromatin, when you can see chromosome, can you see both at the same time? So answer for this. Next, what are the differences? What are the differences between chromosome and chromatin? Differences between chromatin and chromosome. Okay, if you know the answer for any one of these three questions, type it in the comments, students. You are going to get the answer in this session for these three questions. Okay, at the end of the session, we'll write down the answer for these three. Okay, so first is block 1. In this, I have mentioned here as prokaryote versus eukaryote. Okay, the ribosome organelle in both is compared. The ribosome organelle in prokaryote and eukaryote is compared here. Ribosomes, so before comparing, it is called as universal organelle. So this is one important MCQ. It can be asked in the Neet as like the organelle which is present both in the prokaryote and eukaryote. One is, the answer is ribosomes, because in prokaryote you don't have any membrane bound organelles. Membrane bound organelles are absent in prokaryote, isn't it? It is seen in only eukaryote, but prokaryote contains ribosomes and eukaryote also contains ribosomes. So this is called as universal organelle. It is present both in the prokaryote and eukaryote. Now the question is it a membrane bound or non-membrane bound? It is a non-membrane bound structure. Now I am going to ask you, what is the shape of this? What is the shape of this? I told you one example, sugar crystals at your home. When you look at the sugar at your home, you should remember, till the exam is over, you should remember it as ribosomes, granular structures. Otherwise, whatever structure you look at which has a granular texture, you should remember ribosomes. So when you are studying for the exam, you should become the exam. Okay, so ribosomes are granular structures. They are non-membrane bound. They are seen both in the prokaryote as well as eukaryote. So you call it as universal organelle. The organelle which is present in both prokaryote and eukaryote. Membrane bound organelles are absent in prokaryote. It is present only in the eukaryote, a well-developed cell structure. Next, you know ribosomes are made up of two subunits. One is larger subunit and the other one is smaller subunit. Okay, what is the main function of ribosome? What is the main function, students? They supply protein for the cell. Protein for the cell. For example, we take protein for function, right? For our muscles, we take protein, isn't it? You can see people working in the gym, they mention some protein, they take protein because it helps in building up the muscles. So each and every cell, this is as a whole body. When you take the small portion, the cell, because we are made up of trillions of cells, each cell require protein. So these proteins are synthesized by ribosomes. So the proteins or the cellular protein need is supplied by ribosomes. These ribosomes are made of two subunits, the larger and the smaller. Now this one already we saw. If you know the answer, type it in the comments. The prokaryote ribosomes are of 70S type. That is 50 plus 30 is 70S type. What stands for what? Denotes S. If you know the answer, type it in the comments, students. We already saw it. Swedberg's unit. Swedberg's unit. See, I won't reply in the middle of the chat, students, because we have dedicated only half an hour, 30 minutes for this session. So we have to focus only. So at the end of the chat, later it's over, I'll reply for all your queries, okay? But you can type it in you can ask your questions, your doubts, or post the answers, or post what subject, what chapter I have to take next, the difficult one, concept based. I'm ready to do it for you, okay? So now coming back to this part, eukaryotes are made up of 60s and 40s, totally 80s, okay, 80 subunits, Swedberg units, okay? It is 60s plus 40s, it's 80s type. Eukaryotes ribosomes are of 80s type, 70s and 80s. This is very important. These values are important. At the same time, these values are also important. It can be asked in the match the following. Here also see 60 plus 40. You should, if you add, it becomes 100. You shouldn't add because they are Swedberg's units. What is the meaning of that? It is sedimentation coefficient. Sedimentation coefficient, which is the indirect measure of what? Measure of density and size. Density and size. Okay. Now, the ribosomes are present in other organelles also. Okay. Ribosomes are present in cytoplasm. We know. Ribosomes are present openly in the cytoplasm. They don't have membrane. We saw it. Now, ribosomes are present in other organelles of the cell. What are the organelles? This one also we studied last week. Name the two semi-autonomous organelles. Very important. Mitochondria and chloroplast. Why are they called semi-autonomous organelles? Because they have their own DNA and ribosomes. We studied, right? Ribosomes. They have their own ribosomes, which means ribosomes are present in mitochondria and chloroplast. So this is the first organelle. Second organelle. What is the third organelle? Rough endoplasmic reticulum. There is a picture in NCERT, that is, you can see the nucleus, the nuclear envelope, the outer envelope, like it will be folded, coming out like this, small crystals, okay? That granular structures, they are ribosomes. So the outer nuclear envelope extends, okay, as endoplasmic reticulum. You can see the ribosomes attached to the endoplasmic reticulum. Tomorrow I'll show in the book, okay? So the third organelle is rough endoplasmic reticulum. I told you know the importance of studying the diagrams. If you can remember the diagram, you can choose this also, this right option, okay, in the exam time. So the three organelles which contains ribosomes are mitochondria, chloroplast and rough endoplasmic reticulum. Chloroplast are present only in plants. Now shall we write 3 times? That is our motto, right? Mitochondria. What are the 3 organelles which contains ribosomes? Chloroplast. All these are given in your NCERT. I have handpicked everything for you. You can go and check this. Okay, rough endoplasmic reticulum. What are the 3 organelles, students? Mitochondria, chloroplast and rough endoplasmic reticulum. Are you writing or are you following me up, students? Mitochondria, chloroplast and rough endoplasmic reticulum. See, writing 3 times saves you a lot of time. Because, see, I want to tell you one thing. In NEET exam, you have seen students saying they took 2 years, 3 years to clear NEET, isn't it? So they have studied first time, second time, third time. Now we are doing the same 3 times recall at a stretch in half an hour. This I would like to mention the importance of this 30 minutes. Okay, I want you to write it down 3 times without any excuses. Students, are you following me up, students? So in this we saw about ribosomes, prokaryote and eukaryote. It is a universal organelle, non-membrane bound, 50s and 30s, 70s type of seen in prokaryotes, 80s is seen in eukaryotes, 60s plus 40s, and S is a Swedberg unit. Organelles which contains ribosomes or mitochondria, chloroplast, which is seen in plants, and rough endoplasmic reticulum. They are also present in the cytoplasm. What is the function of ribosome? It supplies protein to the cell, that is protein synthesis of the cell. Then I asked you, right? What question did I ask you? Chromatin and chromosome are both the same. Now you will have the, after listening to this, you will have the answer. You will know the answer, okay? See, in the nucleus, in the cell, you have cell. This is the nucleus. Right? Inside this, you have, what are all the structures present inside the nucleus, students? Come quick, type it in the comments. You have nucleoplasm. First, always remember a liquid. In the cell means there is a liquid. In that, organelles are fixed, that is glued, okay? So, organelles, same inside the nucleus also. Here you have cytoplasm. Here you have nucleoplasm. Nucleoplasm, this is a nucleus is a double membrane bound organelle. It has two layered covering, double membrane bound. There are openings here, okay? You call it as nuclear pores. Nuclear pores, because there should be communication, no? Between the nucleus and the other organelles of the cell. They are all working together, so there should be communication here and there. You call it as nuclear pores. Now the question is whether it is unidirectional or bidirectional. Is it only one way or two way? If you know the answer, type it in the comments, okay? Now inside the nucleus you have what? Genetic material, DNA. There is net. See here, this one also we saw yesterday. Nucleoplasm contains what? Nucleoli, spherical structures called as nucleoli, which synthesizes rRNA. Plus you have chromatin. See this word chromatin contains what? Chromatin. So what is the meaning of chromatin? Chromatin is a, wait, I'll adjust the focus. Chromatin is network of nucleoprotein, okay, like this loose network throughout the nucleus. It is like a loose network filling the nucleoplasm, okay? That is called as chromatin. Chromatin was, now what is chromosome? I explained you chromatin. What is chromosome? See, for that you have to understand the stages of the cell. Actually the answer is both are same. Both are same. Chromatin and chromosomes are same. So for better understanding, I am going to explain you with an example. Okay. See, you have clothes. Okay. You are staying at home. Now the clothes are not arranged properly. They are messy. Suppose you are moving to hostel. What did you do? You pack all these clothes tightly so that you can travel it easily. Okay, same way both chromatin and chromosomes are same. When the cell, this chromatin is seen during the resting phase of the cell. That is it is placed loosely all over the nucleoplasm. Now when it enters cell division, what it does is the chromatin is tightly packed into chromosome. The same chromatin, it changes only what? The external appearance, okay? It gets tightly packed into chromosome. So both the compositions are same. I told you know whether there is any difference between these two. No, the compositions of both the chromatin and chromosome are same. Both are same only. You can see chromatin. Can you see both at the same time? No, it is not possible. Chromatin can be seen only when the cell is in rest. When the cell enters cell division, you cannot see chromatin. Okay, it gets packed into the form of chromosome. See here, now you can understand chromatin versus chromosome. It is a nuclear protein network. I told you know what is given in your NCERT book. You can see chromatin, the loosely packed. In the cell chapter, they have mentioned it as loosely packed network, nucleoprotein network. So what is the composition of chromatin? You can, for students, now we'll move on to the block 3. That is, this side you have, I mentioned the chromosome name based on the centromere position and the shape. The centromere position looks like a particular alphabet shape. Okay, so these 3 are important for the exam. Now first is depending on the centromere position. I told you the centromere position. What is the meaning of centromere? There is a constriction at the chromosome. This constriction is called as centromere. This constriction is called as centromere. Constriction of the chromosome is called as centromere. Now, if the constriction is at the center of the chromosome, you call it as metacentric. Exactly at the center means it is called as metacentric. It is, that is in the middle. Okay, now you have 2 equal arms. See this equal arms. And how does it look like? V shape. So this shape is V. Next is submetacentric, which means a little away from the middle. A small few inches above, okay, from the middle. That is slightly away from the middle, which results in 1 short arm and 1 long arm. This is submetacentric. Sub means below, okay. So metacentric, it is away few distance, it is away, so you call it as submetacentric. The shape is L shape. Next is acrocentric. It is close to its end, okay, almost close to the end of the chromosome, but it's not at the end of the chromosome. So results in 1 short arm, which is very short, and one extremely short and 1 very long arm. This is J shape. Acrocentric is J shape. Next is telocentric. In this, the constriction is almost at the end. Okay, the centromere is almost at the end. That is terminal. Terminal means what? Dead end. Termination. Terminal means dead end. It is almost at the end of the chromosome, which results in a rod-like shape or I shape. Okay, so metacentric. What are the 4 types? Based on the centromere position, you have metacentric, which is in the middle. You have 2 equal arms. Next is submetacentric, which is few away from the center. You call it as submetacentric, slightly away from the middle, 1 short and 1 long arm. Next is acrocentric, which is close to the end but not at its end, 1 extremely short and 1 very long arm. Next is telocentric, which is at the terminal, at the final end. So metacentric is, when you look at this, it coincides with the letter shape, okay, alphabet shape. Metacentric looks like V shape. Submetacentric looks like L shape. Acrocentric, J shape. And telocentric is I shape, that is rod shaped. This can be asked as the match the following. Match the following. Okay. They can ask you directly the shapes, or they can ask you this one, the centromere position and how the chromosome appear, whether they have equal arms, submetacentric, slightly away from the middle. So they won't give this one, they will give this as the answer. 1 short and 1 long arm. You have to find out it is submetacentric. If the centromere is at the end, you have to mark it as telocentric. Okay, this is important for match the following. Now students, shall we write it 3 times? What are the 4 types? Metacentric, submetacentric, acrocentric and telocentric. Now the second time when we are going to write, we are going to repeat this also. Metacentric, in the middle, the centromere is in the middle with equal arms. Next, submetacentric, that is moving away, it has moved away from the middle, one short and one long arm. Next, acrocentric, which is close towards the end, one extremely short and one very long arm. Telocentric, that's terminal at the end, okay, which looks like a rod. Again, third time we are going to repeat with the shape. Okay, metacentric, what is the shape? V shape. Submetacentric is L shape. Acrocentric, J shape. Telocentric is I shape. Okay, so students, are you able to get the points? Did you follow me? Did you follow me, students? I want your feedback. Even it can be your doubt or your answer or discussion. You are always welcome. Today I am going to share one important hack about Neet. That is, after this we'll do the revision, final wind up. Okay, so Neet exam. Neet exam tests your what? Tests your what does it test? Your talent, memorization, okay, talent, memorization. Which one do you think they? Intelligence. Yeah, I forgot to mention intelligence. Do you think the exam tests your talent or memorization capacity or intelligence? Definitely not. This exam tests your analytical thinking. Analytical thinking, choosing the right one. Okay, analytical thinking and critical reasoning. You have to understand the exam pattern, then only you can clear the Neet exam. Before fighting a battle, we have to know what we are fighting for, right? Then only we can frame the steps to win the battle. Okay, so if you think the exam tests your talent, wrong. Everyone is talented, students. Memorization capacity, everyone can memorize. It's only about focus and repetition. That is not a issue. Intelligence, everyone is born intelligent. No one is lesser to anyone. Okay, you all need to understand this, because sometimes when we lose our marks and one of our friend gains more mark, then we'll feel dejected. We think that we are not capable or we are not intelligent. That's not at all true. Okay, I want you to understand that Neet exam tests your analytical thinking and critical reasoning, because this is what a doctor needs. This is what a doctor needs. Okay, actually Neet is your pre-training to be a medico. A pre-training to be a medico. So start reading Neet, the NCERT, as a conceptual based one. Don't do memorization. Write, okay, write everything, draw, understand the concepts. Okay, even if there is no drawing, from what you study, create some drawing and do the study. Okay, so from now onwards you have to study NCERT based on the analytical thinking and critical reasoning. That is why they are asking you type of questions, isn't it? Otherwise they can ask question. Okay, because this is very important for a doctor. Now we'll do the final wind up after revising the students. Are you following me, students? Are you following me? Okay, I want your interaction in the chat, students. Even after the session is over, if you are watching a replay of this, kindly comment me in the comment section. That will be helpful for me to do to decide what chapters I have to do next and what are the refinements I have to do in my session, in this half an hour session. Okay, we can do a live flow chart also. If you are interested, you can mention the cycle, okay, Krebs cycle or the pathway, respiratory system pathway. You can mention it. We'll do it on one particular day. Okay, so block 1, prokaryote versus eukaryote. Both contains ribosomes, universal organelle, non-membrane bound, made up of two subunits. That is prokaryotes has got 70s and eukaryotes 80s type. 70s is made up of 50s and 30s, small, larger and smaller subunit. 60s and 40s, small, larger and smaller subunit. Next, the organelles which contain ribosomes are mitochondria, chloroplast and rough endoplasmic reticulum. It is also present in the cytoplasm. The function is protein synthesis for the cell. Next, you have nucleus. Now, you got the answer for all this, right? Yeah, now you have to tell me. Chromatin and chromosome are same. Chromatin and chromosome cannot be seen at the same time in the cell. So when can you see chromatin? In the resting phase, that is the interphase, when the cell is not dividing. Chromosome, the same chromatin appears as chromosome in cell which undergoes cell division. That's all. Okay, there is no difference. Both are same. Both have same composition. Same composition. They just change their appearance. That's all. Okay. Next, chromatin versus chromosome. Both are nucleoprotein network, nucleoprotein complexes. The appearance, there is a difference in appearance. When I say there is no difference, don't think there is no difference. Okay, I mentioned the composition, but the appearance wise they are different. This is loosely packed, this is tightly packed. The composition is same for both. Okay, so same person dressed in a different way, two different way. That's all. DNA, histones, non-histone and RNA. Histones are basic proteins. Okay, so DNA, histone, basic proteins, non-histone proteins and RNA. This is the composition of chromatin and chromosome. See this, there is no diagram for this. From the NCERT, with the understanding, I have made one diagram like this. I want you to do like this. When you study, create a diagram, your own diagram. Okay. Cell, nucleus, when the cell is in the resting phase, interphase, you can see nucleoprotein fibers, that is the chromatin. Once the cell enters cell division, you cannot see chromatin or nucleus. Both are absent. Important MCQ. The structure which is absent when the cell undergoes cell division or nucleus and chromatin, because chromatin gets condensed into chromosome. So when can you see chromosome? Visible only in the dividing cell. Okay. Students, did you get this points? Next is chromosomes based on the centromere position: metacentric, submetacentric, acrocentric and telocentric. This constriction on the chromosome, you call it a centromere. If the centromere is at the center, the middle, it is called as metacentric. It has got equal arms and it looks like V shape. When the constriction is slightly away from the middle, it results in one short and one long arm which looks like L shape. Then acrocentric. This one is, the centromere is very close to the end, but it is not at the end, resulting in one extremely short and one very long arm. That is J shape. Telocentric. Terminal, that is here the centromere is at the dead end, the final end. You call it as telocentric, which looks like I shape or rod shape. Now the question is, which one is P arm and which one is Q arm? Type the answer in the comments. Okay, hope you enjoyed this session. I really enjoyed this, and I request you to practice this. Even when you do watch it in replay, write it as many times as possible, and stay tuned with the Neet Cognition students. Thank you for staying patiently with me. We'll meet you in tomorrow's session at 5 o'clock live. Thank you.